Hello guys, today we are going to start this chapter which is excretory products and their elimination. This is from the unit Human Physiology of class 11th NCERT. So without any further ado, let's begin this chapter. We all know that all the animals, they accumulate different toxic waste in their body by either metabolic activities or by any other means like excess ingestion, for example. They all accumulate these waste, and we all know these wastes are very harmful and these wastes are toxic for our body, so they have to be removed either totally or partially. Now why partially? Because certain wastes like urea, for example, in our case is also important for conservation of water. So basically urea plays important role in conservation of water. So certain part of urea in some of the organisms like humans also is retained in kidney matrix, actually for maintaining the osmolarity in order to conserve water. I've just told you in short what is countercurrent mechanism. This is the whole mechanism that we'll read in this chapter only. In the later topics about the conservation of water in urea. But moving on, all these toxic substances have to be removed totally or partially. For that we need mechanism of elimination, and that is what we are going to study in this chapter, with special emphasis on common nitrogenous wastes, which are ammonia, urea, uric acid. These are major nitrogenous wastes excreted out by the bodies of animals. Ammonia is the most toxic form and requires large amount of water for the elimination, whereas uric acid is the least toxic form of nitrogenous waste and requires minimum amount of water for its removal, which means it can be removed with minimum loss of water. Now, ammonotelism, the process of excreting ammonia. Ammonotelic in nature, ammoniotelic animals means the animals or organisms which excrete ammonia is known as ammonotelic in nature, which is bony fishes, aquatic amphibians and aquatic insects. So bony fishes, aquatic amphibian, aquatic insects, all these live in water. They are all aquatic. Why? Because ammonotelic, or basically ammonia removal, requires large amount of water, so they have to be aquatic. Now ammonia is readily soluble, which is very much evident in the fact that it requires large amount of water for its removal. So ammonia is readily soluble in water and it is excreted by diffusion through body surface or through gill surfaces in fishes. That's ammonium ion. So what is there? Basically they excrete ammonia in the form of ammonium ion over their body surface, and that is easily removed out into the surrounding from their body by simple mechanism or simple physical process, which is diffusion. So there is no role of kidney in its removal. Another statement, whatever statements I have highlighted with this color are important for statement based type of questions. Now terrestrial adaptation necessitated the production of lesser toxic nitrogenous wastes like urea, uric acid for conservation of water. What does it mean? Basically terrestrial adaptation require production of urea and uric acid because these are lesser toxic wastes and these require less amount of water for its removal. And for terrestrial adaptation we need water conservation. So for that matter terrestrial adaptations have to conserve water in order to live over terrestrial region, right? So for that matter terrestrial adaptation necessitated lesser toxic nitrogenous waste production. Then we have ureotelic animals. Ureotelic animals are mammals, terrestrial amphibians, marine fishes. All these are ureotelic animals. And how urea is formed? Urea is formed by ammonia conversion, which means ammonia, first of all, which is produced by metabolism, is converted into urea in liver of these animals. Then it is released into the blood, and from there it is removed by kidneys. Now here kidneys play important role in the removal of urea because urea cannot be physically removed out of the blood of that organism into outside by any simple process like diffusion, right? So kidneys are required here for the removal of urea from the blood. So first of all blood is filtered by kidney and then urea is excreted out of the body by kidneys. This is the statement which I was talking about. Some amount of urea may be retained in the kidney matrix of some of the animals to maintain desired osmolarity. Now, why we want to maintain desired osmolarity? Because we want to conserve water, and for conservation of water, we have to maintain desired osmolarity. We'll talk about it in detail in the upcoming topic of this chapter only, which is countercurrent mechanism. So don't worry about that. And then we have uricotelic animals, which remove uric acid from their body. Examples are reptile, birds, land snails, and insects. They remove uric acid in the form of pellets or paste, and they require very less minimum amount of water for the removal of these waste. So that's good. Then we have different excretory structures present in different animals of animal kingdom. Invertebrates have simple tubular forms of excretory structures, and vertebrates have complex tubular organs for excretion like kidneys. Now let's talk about some of the structures present in different types of animals in case of animal kingdom. So protonephridia or flame cells are present platyhelminthes, also known as flatworms. Example is planaria. Rotifer, some annelids. Cephalopoda, example is amphioxus. All these have protonephridia or flame cells. Protonephridia is specially or primarily concerned with ionic or fluid regulation, which is osmoregulation. This statement becomes important because it's written that it's primarily concerned with only osmoregulation. If now in examination examiner trick this question and change the statement as protonephridia is primarily concerned with let's say removal of nitrogenous waste, then that statement would be a false statement, because in NCERT it's clearly mentioned that primarily it is concerned with osmoregulation. That's it. Then we have nephridia, which is another excretory structure, again tubular excretory structure, which is present in earthworm and other annelids. Now nephridia help in removal of nitrogenous waste and also help to maintain ionic balance or fluid balance, which is osmoregulation. So nephridia is also important for both of these functions, which is removal of waste and osmoregulation. Then we have Malpighian tubules, which are excretory structures in most of the insects like cockroaches, and they also help in removal of waste, nitrogen waste, and osmoregulation both. Remember the word Malpighian tubule because there is one more upcoming word which is Malpighian body, which is different from this. We have another excretory structure which is antennal glands or green glands. They are present in crustaceans like prawns, and they also play important role as they perform the excretory function in these prawns, right? And then we have human excretory system. Human excretory system consists of pair of kidney, a pair of ureter, one urinary bladder, and one urethra. Now kidneys are reddish brown bean shaped structures which are present between T12 to L3. This is very important question which is directly asked. Basically the location of kidney is between T12 to L3. For example, this is kidney, then it is present between T12 to L3. Now what do these mean, right? So for that we need to study a little bit about vertebra, different types of vertebra. So basically we are going to study this about in detail in case of locomotion. However, let me tell you about these. We have total 26 vertebra, 7 cervical vertebra, 12 thoracic vertebra, 5 lumbar vertebra, 5 fused, which means 1 sacral vertebra and 1 coccygeal vertebra. Now 5 close bracket, which means 5 bones fused together to form 1 sacral vertebra. Similarly 4 bones fused together to form 1 coccygeal vertebra. Talking about the location of kidney, kidney is located between T12 and L3, which means 12th last thoracic vertebra and 3rd lumbar vertebra, so L3, and it is close to dorsal inner wall of abdominal cavity, which means it's close to dorsal inner wall. If they trick the question and change this word, like they might change this into ventral inner wall or ventral outer wall, then that would be incorrect. Kidney dimension is 10 to 12 cm in length, 5 to 7 cm in width, 2 to 3 cm in thickness, and average weight is from 120 to 170 gram. And this is the human urinary system. We have, first of all, we have 2 kidneys, a pair of kidney, 2 ureters, a pair of ureter, then you have urinary bladder, and lastly you have urethra. One more thing to note here is that we have adrenal gland on the top of this kidney, so they are also known as hats of kidney because they are present in form of like hat of kidney, right? I hope you understood. Now, talking about these statements, which are very important to understand the section of kidney. So basically, towards the center of inner concave surface of kidney, there is a notch called hilum. So for example, this is a kidney. So towards the inner concave surface, this is the inner concave surface, we have a notch. This is called hilum, where ureter, blood vessel, nerve enter. So for example, ureter, blood vessel, nerve, all these things enter from here. This is a notch which is called hilum. Now inner to the hilum, we have broad funnel shape which is the renal pelvis with projection called calyces. So inner to this we have renal pelvis, and renal pelvis has also certain projections which are called calyces. So these are called minor calyces, major calyces basically, which are basically inner to the renal pelvis are the major calyces. So they are major calyces. Then talking about outer layer of the kidney is a tough capsule, and inside the kidney we have two zones, outer cortex and the inner medulla. Medulla is made up of conical masses, divided into conical masses which are called medullary pyramids, projecting into calyces, and the cortex extends in between, that we'll talk about later. But first of all, medulla has medullary pyramids. So outer surface is cortex, then we have medulla where you have triangular shaped medullary pyramids. They project into minor calyx first, these are the minor calyx, then these are the major calyx, then you have renal pelvis, and then you have hilum, right? Cortex also extend in between these medullary pyramids, and that region of cortex which penetrates into these medullary pyramids, that are called renal columns, and they are called columns of Bertini. Now each kidney has 1 million complex tubular structure. So two kidney will have 2 million complex structure which are nephron. I'm just saying this because sometimes they directly ask how many nephrons are present in total kidneys or in both kidneys. Then we kind of think that they're asking about one kidney, but they were actually asking about two kidneys. So just reminding you, such questions can also be framed and they can also trick on you. Then we have each nephron is composed of two parts, so glomerulus and renal tubule. First of all, let's talk about glomerulus, then we'll talk about renal tubule. Glomerulus is a tuft of capillary which is formed of afferent arteriole, which is a fine branch of renal artery, and blood from the glomerulus is carried away by efferent arteriole, which is again a fine branch of renal artery. So first of all, we have to understand, let's understand it with the picture, that glomerulus is tuft of capillaries made up of afferent arteriole, which is again a form of renal artery, and the artery which comes out of it is efferent arteriole. Okay, it's efferent arteriole, which is again a fine branch of renal artery. Then we have renal tubule. Renal tubule begins with double-walled cup-shaped structure. It's called Bowman's capsule. It encloses the glomerulus, and glomerulus together with Bowman capsule is called Malpighian body. This is what I was talking about. Malpighian body is different from Malpighian tubule. Malpighian tubule is simply the structure which was present most of the insects and cockroaches. However, Malpighian body is basically glomerulus along with Bowman's capsule. Now after Bowman's capsule, renal tubule begins with Bowman capsule, but after that we have tubule continues and further forms highly coiled network, which is proximal convoluted tubule, and then from there we have hairpin structure which is called Henle's loop, which has both descending and ascending limb. Ascending limb again continues to form highly coiled tubular region which is called distal convoluted tubule. Now DCTs, which are distal convoluted tubule of many nephron, open into straight tube which is called collecting duct. So different distal convoluted tubule open into one collecting duct, and then there's several collecting duct as well, and several collecting duct, basically several DCT first of all open into one collecting duct, and there's several collecting duct in similar fashion, and then these collecting duct then combine together and open into renal pelvis, and they open into renal pelvis by crossing first of all medullary pyramid, then calyces, then renal pelvis. So open into renal pelvis through medullary pyramid and calyces. I'll tell you the whole sequence how urine is like transported and stuff, but let me first of all talk about the renal tubule first. So efferent arteriole enters into this Bowman capsule, and this forms tuft of capillary which is called Bowman capsule, and efferent arteriole exits from here. That is the first part of nephron. Second part is the renal tubule, which is composed of one Bowman capsule, which begins from this cup-shaped Bowman capsule, which comprises tuft of capillaries called glomerulus. This is the Bowman capsule, and this Bowman capsule basically continues as coiled structure, and this coiled structure is called PCT, and this PCT then forms a hairpin like structure, which is called what, which is loop of Henle. So there is descending loop of Henle, ascending limb of Henle, right? Ascending limb of loop of Henle, descending and ascending, they are both two loops, right? Now after that, the ascending limb also form a coiled structure, which is called distal convoluted tubule, and then several DCTs, distal convoluted tubule, open into one collecting duct, and the several collecting ducts also, they combine together to form next substance, like they combine together and then urine is transferred from the collecting duct to medullary pyramids, then into minor calyx, then major calyx, and renal pelvis, and then from there it is transported to ureter. We'll talk about it. So let's actually talk about it from here. So we have nephrons. Nephrons enter into, like we have collecting duct, they enter into this medullary pyramids, and then from there medullary urine enters into minor calyx, right, like this, and then into major calyx, and then finally into this renal pelvis, then into ureter, like this, and then it moves into urinary bladder, urethra. I've written here also, urine first of all moves from, not urine exactly, urine name is given to the filtrate only after it's collected in collecting duct. So here it is filtrate, then filtrate goes into PCT, then loop of Henle, then DCT, and from here urine is formed, and urine from collecting duct goes into minor calyx, major calyx, renal pelvis, ureter, urinary bladder, and then urethra. I hope it is clear, right? Now let me show you the figure of kidney, which is this. Actually I wanted to show you the minor calyx structure. So the structure which is U-shaped here, right? So these are not minor calyx. The portion which is in between them, this is the minor calyx. You can see these are the minor calyx. These are the minor calyx, and then they combine together form this, which is major calyx, and then major calyx, and then you have this structure which is funnel shaped. This is called what? Renal pelvis. This was not clear in the NCERT, so I wanted to show you. This is not calyx. The structure between them is calyx, which can, between them is calyx, which can easily be seen in the figure, right? Then let's talk about the next paragraph, which says that Malpighian corpuscle, which is Bowman capsule plus glomerulus, PCT, DCT of nephron, all are situated in cortical region of the kidney, whereas loop of Henle dips into medulla. So I have to mark this, very important, that if we talk about nephron, then parts of nephron, then we have this Bowman's capsule together with tuft of capillary in it, which is glomerulus of course, and then we have PCT, then we have loop of Henle, and then we have DCT. So Bowman capsule, DCT and PCT, they all are located in cortex, whereas loop of Henle is located where? In medulla. So loop of Henle is located in medulla, but there are two types of nephrons. Let's talk about that. In majority of nephrons, loop of Henle is too short and extend only very little into the medulla. See, loop of Henle extends into medulla, but if it extends only very little, right, then in that case they are called cortical nephron. If in case that loop of Henle is very short and it extends only very little into the medulla, then they are called cortical nephrons. But in some other nephrons where loop of Henle is very long and runs deep into medulla, then they are called juxtamedullary nephrons. That is the difference. But anyways, we have to remember this for statement made questions, that Malpighian corpuscle, which is Bowman capsule plus glomerulus, PCT, DCT, all are situated in cortical region, whereas loop of Henle dips into the medullary region. important. Then we have efferent arteriole emerging from the glomerulus form fine capillary network around renal tubule. So this is the whole renal tubule, right? Bowman capsule, PCT, then you have loop of Henle, then you have DCT, and then several DCT opens into the collecting duct. That's different point, but this whole structure is one nephron, right? And of course I have told you that CD, which is collecting duct, is not a part of one nephron, right? Several nephrons DCT opens into CD, so it's not part of one single nephron. So what are they saying? Efferent arteriole which is emerging from the glomerulus. So efferent arteriole was coming into the glomerulus, and then glomerulus was forming tuft of capillaries, and efferent arteriole was like, let's say this the efferent arteriole. This efferent arteriole was coming out from the glomerulus, right? And that forms fine capillary network around renal tubule. So around this whole renal tubule, a capillary network, fine capillary network is formed, and that is called peritubular capillary. And from that peritubular capillary there is a minute vessel of this network which runs parallel to the loop of Henle, forming U-shaped vasa recta. So that is forming U-shaped vasa recta, which means, I'll show you with the help of a picture. So you can see this is the whole capillary network. This is called peritubular capillary. Now what is this? First of all, efferent arteriole enters into Bowman's capsule, and then there's tuft of capillary which is formed because of efferent arteriole, and then efferent arteriole comes out of it, and efferent arteriole then forms this capillary system around renal capsule, around renal tubule. This is the whole renal tubule, right? This is the whole renal tubule. So around this renal tubule, we have capillary system which is called peritubular capillary. That's the point. And this part of capillary, peritubular capillary, which is parallel to the loop of Henle, this is called vasa recta, and this is of course U-shaped. Now next point is vasa recta is absent or highly reduced in cortical nephron, which means first of all, cortical nephron are those nephron where loop of Henle is very short, it only penetrates very little into the medulla, and secondly, vasa recta is highly reduced in cortical nephron, whereas in juxtamedullary nephron, loop of Henle is very long and runs deep into the medulla, and also vasa recta is not reduced, it's present in these types of juxtamedullary nephrons. Got it? Then next point is urine formation. That's the most important topic from this chapter, which is urine formation. It involves three main processes, which is glomerular filtration, reabsorption, and secretion. It takes place of course in different parts of the nephron. Now first step in urine formation is filtration of blood, which is carried out by glomerulus, therefore it is called glomerular filtration. This is a very important statement that on an average 1100 to 1200 blood is filtered by kidney per minute, which constitute 1/5 of the blood pumped out by each ventricle in the heart in a minute. So each ventricle in 1 minute pumps how much? 5000 ml of blood, and this is approximately 1/5 of the 5000 ml of blood which is pumped by one ventricle, each ventricle in a heart. And this amount of blood is filtered by kidney in 1 minute. I hope you got it. So basically 1100 to 1200 ml of blood is filtered by kidney in 1 minute, and 5000 amount of blood, 5000 ml of blood is released or pumped by each ventricle of heart. So what is the connection between the two? That 1100 to 1200 ml of blood is 1/5 of the blood which is pumped out by each ventricle, which is 5000. Now next part is that what is the cause of this filtration? First of all, it is done by glomerulus. That's nice. This fact is nice. Then what is the cause of this filtration? So the main cause as per the NCERT is glomerular capillary blood pressure. So because of this pressure, filtration of blood happens, and it happens to 3 layers. What are these 3 layers? First is endothelium or Endothelium is basically epithelium of any blood vessel. So endothelium of glomerular blood vessel, epithelium of Bowman capsule, then basement substance between these two layers. These three layers form filtration layer, and this filtration layer is basically three layers from where through where blood passes and get filtered. Okay, and the main reason for the filtration is glomerulocapillary blood pressure. Let me show you here that first of all, efferent arteriole, diameter of efferent arteriole is more, right? And then we have lesser diameter of efferent arteriole. This is going to create a pressure, and according to NCERT, the main cause of glomerular filtration is glomerulocapillary pressure. So what happens is efferent arteriole is entering and it is forming tuft of capillary, which is glomerulus, and efferent arteriole is coming out of it, and this is the Bowman capsule which is present. Now there are three layers through which this blood is going to pass, through three layers, and then it is going to enter into this lumen of Bowman capsule, and then it's going to get transported into all the different parts of the renal tube, which is PCT, loop of Henle, DCT and all. So first of all, the blood here is going to be passed through, first of all, the endothelium of glomerular blood vessel, then through basement substance of glomerular blood vessel endothelium, and the basement membrane of membrane of glomerulus, also basement membrane of Bowman capsule. I'm sorry. So let me actually explain it again. Basically, there are three layers through which the blood is going to pass from the glomerulus into finally the lumen of Bowman's capsule. So first of all, it's going to pass through endothelium, then it is going to pass through the basement membrane of this endothelium of glomerular blood vessel, then it is going to pass through the basement membrane of Bowman capsule epithelium, then it is going to pass through the Bowman capsule epithelium. So this is Bowman capsule epithelium, then you have the basement membrane of Bowman capsule epithelium, and you also have this basement membrane of endothelium, and these two, basement membrane of endothelium and basement membrane of Bowman capsule epithelium, both of these are together known as basement substance. I hope I made sense. Now moving on to the next part, which is epithelial cells of Bowman capsule are known as podocytes, and they are arranged in integrate manner so as to leave some minute spaces, and these minute spaces are called filtration slit or pores. These are the spaces through which the filtered blood, which is also known as filtrate, that enters. Blood is filtered so finely that all the constituent of plasma is there in the filtrate except protein. So all the constituent pass except protein into the lumen of Bowman capsule. And what does plasma have? Which means what does the filtrate has? Basically, I'm talking about what does filtrate have, because I want to mention that all these substances include certain substances which are essential for our body and also include unessential substances. So we have to reabsorb the essential substances, we have to remove unessential or non-essential substances. So just for that, plasma consists of 90 to 92% of water, 6 to 8% of protein, which is not there in the filtrate as it is mentioned except protein, and rest substances are glucose, amino acid and certain salts like sodium, calcium, magnesium. And also one more thing, that plasma also consists of some waste like urea and other nitrogenous waste also. So urea is a major nitrogen. We are urotelic animals, humans. So if we are talking about human excretory system, then we'll be only talking about urea as nitrogenous waste. So this is called ultrafiltration. Why? Because blood is filtered so finely. Why is it filtered so finely? First of all, because it is going to pass through three layers, which is endothelium of blood glomerular blood vessel, the epithelium of Bowman capsule, the basement substance between these two, which consists of the basement membrane of glomerular blood vessel and basement membrane of epithelium of Bowman's capsule as well. So first reason is this. The second reason is that podocytes, which are the epithelial cells of Bowman capsule, have minute pores, minute spaces in between them, which are called filtration slit or slit pores, through which blood is going to get filtered. Therefore, it is filtered so finely, and that is why it is called ultrafiltration. I hope I made sense. Moving on to the next part. First of all, amount of filtrate formed by kidney per minute is called glomerular filtration rate, which is 125 ml per minute. See what happens, 1100 to 1200 ml of blood enters, and then filtered amount is 125 ml only. So out of 1100 to 1200, filtered amount is this, right, which is 180 liter per day. Now we have kidneys have built-in mechanism for regulation of glomerular filtration rate. One such efficient mechanism is carried by juxtaglomerular apparatus. First of all, this mechanism we are going to read in hormonal mechanism, hormonal regulation, which is going to come at the last of this chapter, so you have to wait for that. But let's talk about juxtaglomerular apparatus first. Juxtaglomerular apparatus is special sensitive region which is formed by cellular modification in distal convoluted tubule and the efferent arteriole at the location of their contact. So basically this is modification, cellular modification of distal convoluted tubule and efferent arteriole where they are in contact. So let me show you. If you draw the nephron Bowman capsule like this, let's say, and then you kind of draw the whole PCT, then loop of Henle, and then you have DCT over here. So there is contact between the DCT and efferent arteriole also, which was entering into this. So there is a contact between them, and this contact is basically there is a cell modification of distal convoluted tubule and the efferent arteriole at the point of at the location of their contact. That is going to form juxtamedullary JGA, juxtaglomerular apparatus. There is if there is fall in GFR, which is glomerular filtration rate, that can activate a JG cell to release renin, and this renin will stimulate the glomerular blood flow, and then it will bring it back to normal. How it does it, we'll just talk about it in the later half. Now comparison of volume of filtrate formed per day, which is 180 liter, to that of urine, which is 1.5 liter only. It shows that 99% of filtrate must be reabsorbed in the renal tubule. Then only the total filtrate is 180 liter per day, and the total amount of urine that we produce is 1.5 liter only per day. Of course, that shows 99% of filtrate is reabsorbed. So first of all, the amount of blood that entered was this per minute I'm talking about, and the amount of blood that was filtrate was this per minute, and then by that we can also calculate this also, but that's not necessary. Then we have tubular epithelial cells. Tubular epithelial cells in different segments of nephron perform reabsorption, which is of course the process where we absorb the substance again. So that's called reabsorption by active or passive means. So tubular epithelial does it by tubular epithelial cells do it by active or passive mechanisms. Now substances like glucose, amino acids, sodium, etc. in the filtrate are reabsorbed actively, whereas nitrogenous waste are absorbed by passive transport. That's the importance of it. So you have to remember this: glucose, ammonia and sodium, amino acids and sodium ions are reabsorbed actively, whereas nitrogenous waste are absorbed by passive transport. There is also reabsorption of water that is also occurring passively in the initial segment of nephron. So in the initial segment of nephron there is reabsorption of water. So these are the statements that you have to remember for reabsorption. Okay, then we have urine formation. During urine formation, tubular cells secrete substances like H plus, K plus, ammonia in the filtrate. And why they does that? Why they do that? I'm sorry. Why they do that? Tubular secretion is also important in urine formation because it helps in maintenance of ionic and acid balance of body fluids. That's the reason. And then we have function of tubules. Here we are going to specifically talk about each and every reabsorption and secretion by each and every part of the nephron. Proximal convoluted tubule is lined by simple cuboidal brush border epithelium. Why brush border? Brush border means it has a microvilli in it, and microvilli is present. Why? Because it is important for reabsorption. So PCT has surface area for absorption, reabsorption, and this brush border epithelium increases its surface area. That's it. So it is made up of lined by simple cuboidal brush border epithelium and all essential nutrients first of all. Secondly, 70 to 80% of electrolytes plus water are reabsorbed in this segment. That is why NCERT was mentioning that initial segments of nephron, water is reabsorbed. So PCT is the initial segment where water is reabsorbed together with all essential nutrients, together with 70 to 80% of electrolytes. Then it also help in maintenance of pH and ionic balance. So pH and ionic balance is maintained by, of course, selective secretion of hydrogen ions and ammonia into the filtrate, right? So and first of all, for pH and ionic balance, hydrogen ions and ammonia are secreted into the filtrate. Okay, and also there is absorption of bicarbonate ion from it. So we absorb bicarbonate ion again from it in order to maintain the pH ionic balance. That is what is written for the case of PCT. So for the case of PCT, there is absorption of SCO3 from it in order to maintain the pH. That is what NCERT is kind of meaning here, right? Then we have Henle's loop. Henle's loop is the part of nephron where reabsorption is minimum, and reabsorption is minimum in its ascending limb, and this ascending limb plays important role in maintenance of a high osmolarity of medullary interstitium. Descending limb of loop of Henle is permeable to water but impermeable to electrolytes, so it concentrates the filtrate, and ascending limb is impermeable to water and permeable to electrolyte, so it allows the movement of electrolyte actively or passively, and that is how the concentrated filtrate, which was because of descending limb, passes upward, and once it passed upward, it is diluted due to passage of electrolyte into the medullary fluid, right? So that was about Henle's loop. Talking about DCT plays important role. Why? Because conditional reabsorption of sodium and water takes place in this segment. DCT is also capable of reabsorption of HCO3 minus. Here NCERT is just saying there is reabsorption, but there NCERT was saying there was absorption of this. So it is kind of sensing that absorption of HCO3 is because of pH balance or ionic balance. So PCT absorption of HCO3 minus takes place because it wants to maintain pH and ionic balance. So to maintain pH and ionic balance, absorption of HCO3 minus taking place. But here there is direct reabsorption of HCO3 minus in DCT. So that is overall another topic. Like reabsorption is there because these are essential for our body, so we kind of take it back from the filtrate. Then we have secretion of hydrogen and potassium ions plus ammonia in the DCT, again to maintain ionic balance, sodium potassium balance, and pH balance. Point to remember is that there is conditional reabsorption in sodium and water in DCT only, because if you talk about collecting duct also, so there is nowhere mentioned there is conditional reabsorption. So only in DCT there is conditional reabsorption, and you have to understand this also. Yeah, collecting duct is not a part of single nephron. It's basically it has a lot of DCTs coming into it, where all those DCT open into collecting duct. It's not like collecting duct is a part of a single nephron, no. So that is why, if for example in coming portion of the chapter we are going to read about conditional reabsorption, so in there is only conditional reabsorption taking place in the later part, which is the DCT of the nephron. That's it. There's no conditional reabsorption taking place in any other part of the nephron. Talking about collecting duct, it extends from the cortex of kidney into the inner part of the medulla. Large amount of water can be reabsorbed to produce concentrated urine. This is what we are going to talk about in detail in countercurrent mechanism. Then it also allows small portion of urea into the medullary interstitium to keep up osmolarity. This is also related to countercurrent mechanism. I'll basically talk about this after I'll help you to understand countercurrent mechanism. It plays important role in maintenance of pH and ionic balance also by selective secretion of H plus and K plus ions. Let's look up this is very important figure to understand everything. First of all, we have cortex. In cortex we have PCT, we have DCT. If you talk about juxtamedullary nephrons, then we have a long loop of Henle extending deep into the medullary region, which is what is shown here. Now we have in PCT, all the nutrients are reabsorbed. NaCl means all electrolytes, and 70 to 80% of electrolytes is reabsorbed, and water is also reabsorbed. There is secretion of H plus and sodium ion. Okay, H plus and sodium ion. Here remember, K plus is not secreted, K plus is reabsorbed. Alright, so H plus and ammonia is only secreted here, and HCO3 minus is also absorbed. And why is it absorbed? For pH and ionic balance. In case of PCT only, that's what is mentioned in NCERT. And then we have secretion of in DCT. Mind you, there is conditional reabsorption of Na plus and water, and there is secretion of K plus and H plus. HCO3 minus is also reabsorbed in DCT, that is clearly mentioned in NCERT. Talking about descending limb of loop of Henle, it is permeable to water but impermeable to any electrolyte. And then ascending limb of loop of Henle is permeable to electrolyte and impermeable to water. So because of that, when descending limb of loop of Henle is going downwards, concentration of urine is increasing. When ascending limb of loop ascending limb of loop of Henle is going upwards, then concentration is reducing, which means diluting. In collecting duct, if we talk about, then water is reabsorbed and urea can also be reabsorbed, and small amount of urea can also flow into, basically urea can flow into this is the inner medullary interstitium, so it can flow into inner medullary interstitium. That's the point. And one more thing is there, that selective secretion of H plus and K plus ions can also take place. Mechanism of concentration of filtrate. Mammals have this ability, which is very unique, to produce concentrated urine. Now Henle's loop and vasa recta play important role in this. So they play important role in concentrating the urine. So there is counter current. What do you mean by counter current? If for example the flow of filtrate is upwards in one limb, then other limb will have flow of filtrate in the opposite direction. That's called counter current. So there is counter current between two limbs of loop of Henle. So flow of filtrate is in opposite direction in two limbs of loop of Henle. That forms a counter current. Then flow of blood. This was flow of filtrate in loop of Henle in counter current. Then we have flow of blood in two limbs of vasa recta. Vasa recta is basically part of peritubular capillary. So flow of blood in two limbs of vasa recta again forms one more counter current. Then we have proximity between Henle's loop and vasa recta, that also forms one more counter current. And all of this help in maintain or help in increasing osmolarity towards inner medullary interstitium, right, from 300 milliosmol per liter in cortex to 1200 milliosmol per liter in inner medulla. And this gradient, which means this concentration increasing, is caused due to gradient, and this gradient is maintained and caused by NaCl and urea concentration. Basically NaCl and urea are the electrolytes and salts which are going to increase the concentration. So what happens and how concentration is increased? So whole agenda of concentrating the urine is by increasing the osmolarity. So we need to increase the osmolarity, and then once we increase the osmolarity, for that we need to maintain the gradient. So then only we'll be able to increase osmolarity continuously, and that will allow easy passage of water from collecting tubule. And once there is passage of water from collecting tubule, then of course what will happen? Urine will become concentrated. That's the purpose, right? So we are going to focus on the primary thing, which is increasing osmolarity, and that is done by NaCl and urea with the help of loop of Henle and vasa recta. So NaCl is first of all transported by ascending limb of Henle's loop and exchanged with descending limb of vasa recta. Then NaCl is returned into interstitium by ascending portion of vasa recta. Right, so first of all let me explain the movement of NaCl first. They have drawn it separately, but of course it's going to be parallel. So let's draw, this is vasa recta. So this is descending limb of loop of Henle, ascending limb of loop of Henle. This is ascending limb of vasa recta, and this is descending limb of vasa recta. That is why there is counter current in their proximity, also counter current. So what I'm talking here is that first of all NaCl is removed out, NaCl is given to the interstitium by ascending limb of this loop of Henle, and then it is exchanged with the descending limb of vasa recta, right? And then NaCl passes through this and then finally enters into the ascending limb of vasa recta. From here NaCl is returned back to the medullary interstitium by the ascending limb of vasa recta. I hope I made sense. Just rewind the video. If you don't understand, you will be able to understand it for sure. Then we have urea also. Movement of urea is also there to maintain the gradient. So there is small amount of urea which enter the thin segment of ascending limb of Henle's loop, and then it's transported back to interstitium by collecting tubule. So what happens is that urea, which is coming from collecting duct only, and this urea enters into, this is the thin segment of ascending limb. So it enters into the thin segment of ascending limb of loop of Henle, and then it basically comes again to the collecting duct via DCT. Of course this is the DCT, and then it comes into the collecting duct, and again urea is given out into the inner medullary interstitium. So this is how urea is continuously flowing into the inner medullary interstitium, increasing the concentration, maintaining the gradient, and that will finally concentrate the urine. So the above mentioned described process or transport of substances facilitated by special arrangement loop of Henle and vasa recta is called counter current mechanism. So this is counter current mechanism whose purpose is to increase osmolarity towards inner medullary interstitium, right, to maintain the concentration gradient, right, with the help of vasa recta and loop of Henle. That is called counter current mechanism. It helps to maintain concentration gradient in the medullary interstitium, and you have to talk about like where is it helps in maintaining concentration gradient in the inner medullary interstitium. Presence of such interstitial gradient, right, helps in easy passage of water from collecting tubule, and once water is gone from the collecting tubule, whatever is there, which is the filtrate, is going to form the urine, is going to get concentrated. So that's how human kidneys can produce urine 4 times concentrated than the initial formed filtrate. I hope it makes sense. Alright, and let me show you that paragraph also, which I told you that we'll talk about it after counter current mechanism. This is the collecting duct, and what happens is that large amount of water is reabsorbed from this region to produce concentrated urine. Reabsorb means it is going out from the collecting duct. That's what we have studied. We are going to increase the concentration of osmolarity of inner medullary interstitium by increasing urea concentration, and by increasing the concentration of NaCl over the inner medullary system. Overall, the purpose of us doing these things is increasing the osmolarity of inner medullary interstitium. Once we'll increase osmolarity, then and maintain the gradient, increase osmolarity in the inner medullary interstitium, then large amount of water will be reabsorbed, will flow out from where? Collecting duct. It will flow out from collecting duct, and this will produce concentrated urine. That is what I was talking about. Then we have this segment also allows small amount of urea. Now urea is the one which is used in maintaining, it was helping in maintaining pressure gradient, right? Concentration gradient, I mean not pressure but concentration gradient. So urea was the one which helped in maintenance of the pressure gradient, and that is how we proceeded and finally formed concentrated urine by removing the water or reabsorbing the water from the collecting duct. I hope it made sense. Now moving on to the next portion, which is regulation of kidney function. Now function of kidney can be monitored, regulated by hormonal feedback mechanism, and hormonal feedback mechanism include first is hypothalamus, then JGA, juxtaglomerular apparatus that we were talking about, and to a certain extent heart also. This is what I have written also. Let's read this. This is related to counter current mechanism. So collecting duct allows passage of water in the inner medulla, but that is due to counter current mechanism. Alright. Then osmoreceptor in the body, first of all, are activated by the changes in first of all, this can be asked as a statement based question. So osmoreceptors are activated by what? Changes in blood volume, changes in blood fluid volume, and ionic concentration. So you have to remember this, that osmoreceptor can be activated by changes in which of them, right? If there is loss of fluid, right, if there is excessive loss of fluid, then that can activate receptor, and that can stimulate hypothalamus to release antidiuretic hormone or vasopressin from neurohypophysis. And ADH facilitates water reabsorption from the later parts of tubule. Now here later parts of tubule is DCT only, right? That is what I was saying, that here water is reabsorbed, but it's not reabsorbed due to these hormonal mechanism. It is reabsorbed due to counter current mechanism. That's different, that's going to be due to the concentration of urea and NaCl in the inner medullary interstitium. That's different mechanisms basically for concentrating the urine. But here DCT is referred to as the later part of tubule, and from here only we have water reabsorption, because DCT is the part of nephron, first thing first. Secondly, DCT forms the later parts of the tubule tubule is referred to as the nephron, right? So therefore, and one more thing, that in NCERT conditional reabsorption is only there mentioned in the DCT paragraph, right? So there is no conditional reabsorption in collecting duct. Whatever amount of water is reabsorbed, it's reabsorbed due to counter current mechanism, not due to hormonal feedback mechanism or other things, right? Not due to conditional reabsorptions, correct? So back to the topic, we have excessive loss of fluid activates the receptor, stimulate hypothalamus to release ADH, antidiuretic hormone, which is also known as vasopressin, from the neurohypophysis, which is the posterior part of the pituitary, posterior part of the hypothalamus. Alright, then moving on, ADH facilitates. Now ADH released, it's released due to loss of fluid. Now it's going to somehow increase the amount of fluid in our body. So ADH facilitates first of all reabsorption from DCT, so it's going to increase the amount of fluid in the body, and it also prevents diuresis. Diuresis means excessive loss of water through the urine. So it does not allow uresis, does not allow loss of water through urine, excess loss of water through urine, by facilitating reabsorption of water from the DCT part. Then there is increase in blood volume, blood fluid volume, which is again blood volume only. Body fluid is blood, so there's increase in body fluid volume Once there is increase, then it switch off the osmoreceptors and then it suppresses the ADH release to complete the feedback. So this is the whole feedback. Okay, first of all, excess loss is there, so it stimulated the receptor activated, got activated and stimulated hypothalamus to release ADH then did its job, prevented diuresis and stimulated, facilitated the water reabsorption by DCT, and then because of all these things there was increase in body fluid volume and that switched off the osmoreceptor and suppressed ADH release to complete the feedback. One more special function that ADH performs is ADH can affect kidney function by constrictory effect on blood vessels as well. So there is an increase in blood vessel because of ADH constrictory effect on blood vessel, and that is why glomerular blood flow is increased and GFR, which is glomerular filtration rate, is also increased. Now point is that why is it increasing the blood pressure? It's never related to, if you think in that way, it's not related to excessive loss of fluid, but it's related to one more important thing. When there is excessive loss of fluid in the body, in our brain there is, like for example, if there is loss of water in our excessive loss of fluid, then the most susceptible part of our body, which is the brain, it is very sensitive towards concentration. If there is less concentration, it might shrink. There is more concentration, it might swell. The cells I'm talking about. So there can be changes in the brain cells because of concentration changes of body fluid. So if there is loss of body fluid, the cells of brain might shrink, or there may be some changes, right? So because of that, there is increase in blood pressure, so that body fluid can directly reach, immediately reach to the brain or other susceptible parts, so that there is no damage provided to them. I hope I made sense. Now JGA, which is juxtaglomerular apparatus, if you forgot, then let me tell you, juxtaglomerular apparatus is made up of cell modification in efferent arteriole, and second was DCT at the location of their contact. So juxtaglomerular apparatus is a complex, it has a complex regulatory role. How? If there is a fall in glomerular blood flow or glomerular blood pressure, or GFR, which is glomerular filtration rate, if there is fall in any of them, it activates JG cell, which is juxtaglomerular cells, activate the cell and that release renin. Now renin is of course a hormone. It will convert angiotensin in the blood, present in the blood, to angiotensinogen in the blood to angiotensin 1, then further converting it into angiotensin 2, and then angiotensin 2, being a powerful blood vasoconstrictor, increases the glomerular blood pressure and thereby glomerular filtration rate. So it has a basically constrictory effect on the blood vessels. It's vasoconstrictor, angiotensin 2. Then angiotensin 2 also activates adrenal cortex to release aldosterone. Aldosterone causes reabsorption of sodium and water again from distal part of tubule. And what is distal part? DCT, not CD. Don't think about CD because it's not even the part of tubule. CD is not a part of nephron. Many DCTs open into CD. Remember, it's not part of single nephron. So distal part of tubule means DCT. That's it. So adrenal cortex is activated, not medulla. Cortex is activated, which releases aldosterone, and aldosterone causes reabsorption, and that is why there was written in NCERT. You remember that conditional reabsorption of sodium and what takes place because of this only. Then this is called renin angiotensin mechanism. Now when there is increased blood flow, if there is decrease in blood flow or decrease in glomerular blood flow or GFR and stuff, then renin angiotensin mechanism was followed and stuff. But now if there is increase in the blood flow to the atria of the heart, then it causes increase in atrial natriuretic factor, which is ANF. Now this ANF will cause vasodilation because there was increase in blood flow. It will dilate the blood vessel so that there is decrease in blood pressure. All body always tries to neutralize, neutralize the thing so that everything becomes okay, back to okay and normal. So we have ANF, which is mechanism ANF mechanism, atrial natriuretic factor mechanism, acts as a check on renin angiotensin mechanism. So it's very important statement for, again, the statement based questions. Moving on to micturition. You know what is micturition? Micturition is basically the process of release of urine. That's what is it. Now, urine formed by nephron is ultimately carried by urinary bladder. Now it is stored in urinary bladder. And like, till how much time it is stored? Till the time when voluntary signal is provided by the central nervous system. Now, when will voluntary signal be provided by the CNS, central nervous system? The signal is first of all initiated by stretching of urinary bladder, which is filled with urine. So urinary bladder is filled with urine, it will stretch. Okay, there will be stretching of urinary bladder. Because of that, signal will be initiated. Now, in response to the stretching, stress receptors on the walls of bladder will send signal to CNS. Now CNS, after sending signal to CNS in reply will provide motor messages to initiate contraction of smooth muscle of the bladder and then simultaneous relaxation of urethral sphincter, causing the release of urine, and that release of urine is called micturition. Two things that are very very important to mention here is that, first of all, for the removal or for the release of urine, we require contraction of smooth muscle of the bladder and relaxation of urethral sphincter. So that is what is required for urine release, which is called micturition, and the neural mechanisms causing the urine release is called micturition reflex. Now, adult urine excretes 1 to 1.5 liters of urine per day. Now we have urine formed, whatever the urine formed is light yellow colored watery fluid and slightly acidic. pH can be asked, which is 6, and it has characteristic odor, which is smell, of course. One more very very important thing: 25 to 30 gram of urea is excreted out per day, and urine is 1 to 1.5 liter of urine is excreted out per day. Now, again, statement based question. Various condition can be affected, various condition can affect the characteristics of urine. So, if for example, if person has certain condition known as diabetes mellitus, then it's his or her urine will be affected, characteristic of urine will be affected, and by that we can analyze the urine, and then that will help us to clinically diagnose many kind of metabolic disorders or malfunctioning of kidney. So that's very essential clinically, right? So for example, if there's presence of glucose, it's called glycosuria, the condition, and if there is presence of ketone body, which is ketonuria in the urine, then these are indicators of diabetes mellitus, because diabetes mellitus is a condition which affect the characteristic of urine in this way, that increase the glucose and increase the ketone bodies, etc. So after analyzing it, we can say that okay, clinically it is diagnosed that diabetes mellitus is present in that person, okay, or is a condition that person is suffering through, whatever. I hope you understood. Then there are roles of organs also, other organs also in excretion, which are lungs, liver, or skin, because kidney is not only organ which is going to excrete. There are several other organs also which is going to excrete. Excretion is basically a word which is for as a total, as a total. What is excretion? It's a removal. It's removal of different kinds of waste. It's just not removal of urine. It's just not release of urine. It's total, right? So there are several organs which are involved in excretion. So lungs remove large amount of carbon dioxide, which is 200 ml per minute. Remember this, very very important. 200 ml of carbon dioxide removed per minute by lungs. Significant amount of water is also removed every day by lungs. Now liver, important, is the largest gland in our body, and it secretes bile containing substances, which is bilirubin, biliverdin, cholesterol, degraded steroid hormone, vitamin, and drugs. All of these are basically secreted by liver. Most of these substances are ultimately passed along with digestive waste, but they are also removed. Then we have sweat and sebaceous gland in the skin. First of all, these two glands are present in skin. Then sweat is produced by sweat gland. It's a watery fluid. It contains sodium chloride, which is NaCl, then urea, lactic acid, etc. So sweat contains sodium chloride, NaCl, lactic acid, etc. So these are removed by the sweat produced by sweat gland. But another function or primary function of sweat is cooling the body. But along with that, there's also removal of waste, which I mentioned, like NaCl NaCl or urea, small amount, or lactic acid. All of these are removed together with sweat, but the main purpose of sweat was primary function was cooling. Similarly, sebaceous gland secretes sebum. Main purpose, of course, is to provide oily covering to the skin, but along with that, it secretes other things like sterol, hydrocarbon, waxes. And one more thing, that saliva also removes small amount of nitrogenous waste. So small amount of nitrogenous waste is eliminated through saliva also. Got it? Then we have disorder of excretory system. Disorder related to excretory system, we are going to read about. First of all, we have malfunctioning of kidney can lead to accumulation of urea in case of urotelic organism like us. Urotelic, right? So in that case, this condition is called uremia, which is accumulation of urea in the blood. It's very harmful because urea is toxic waste and it has to be removed, and it may lead to kidney failure. Now in kidney failure patient with kidney failure, urea is removed by the process called hemodialysis. Why? Because kidney is not working, there's malfunctioning in kidney, so there is accumulation of urea in the blood, which is called uremia. So that's very harmful. So in such patients, either the patients die or urea is removed from their body through hemodialysis. It's during the process of hemodialysis, blood, first of all, let me actually mention this, that these lines, that how the hemodialysis takes place, this is the first thing, and second thing, third thing, these are taking place. So these lines can be asked as a sequence type of question, sequence based question, where you'll be asked to put all these things in sequence. So read it carefully. Now during the process of hemodialysis, blood is first of all drained from artery, and it's pumped into dialysing unit, which is called artificial kidney. So there is a dialysing unit. So we take the blood from artery and then we pump it into dialysing unit. And you have to understand one more point, that blood is drained from convenient artery and is pumped into dialysing unit. That's what is happening, right? But it is done only after adding anticoagulant like heparin. So first of all, we take the blood from the artery, then we add anticoagulant in the blood. So we add anticoagulant like heparin, and then we pump it to the dialysing unit. One more thing, that this dialysing unit has a coiled cellophane tube which is surrounded by the fluid, which is dialysing fluid. Now dialysing fluid has same composition as that of plasma, except nitrogenous waste. And the porous cellophane membrane of the tube allow passage of molecule based on the concentration gradient. So if it does not have a nitrogenous waste, then all the nitrogenous waste which are present in the blood taken from the artery of that person after adding heparin, all of that the waste will be removed out into this dialysing fluid, because there is no concentration of the waste in that dialysing liquid put inside the dialysing unit, right? I hope you understood. And because of that, that dialysing fluid is also changing again and again, so that nitrogenous waste can be removed very easily. Now these nitrogenous wastes are absent in dialysing fluid, so these substances move freely out, clearing the blood, and then cleared blood is pumped back to the body through the vein after adding antiheparin. So it's pumped back to vein, and then we have to add antiheparin first of all in it, right? And it's boon for thousands of uremic patients. Uremic means those patients which are suffering from uremia, which is a condition where there is accumulation of a urea because of malfunctioning of kidney. Right. One more thing, that here in dialysing fluid we have same composition of plasma, except that they don't have nitrogenous waste. But if I ask you about one more type of fluid which has same composition of plasma except that it has no protein, then that will be filtrate. Filtrate has no protein and same composition of plasma. You can rewind back the video, I have told you. Then we have kidney transplantation is ultimate method for the correction of renal failures. Kidney failure, again malfunctioning, same thing. Kidney failure is caused by malfunctioning of kidney. When there's malfunction of kidney, there's uremia, which is accumulation of urea, which is of course the toxic base, because kidney is not functioning well, so it cannot remove the urea, and that's how it's accumulating inside, and that's producing a condition which is uremia, and because of that it can lead to kidney failure. Functioning kidney is used in transplantation from the donor, and we have to find a matching donor to minimize the rejection by immune system of the host. So we will choose a close relative. Now, modern clinical procedures have increased success rate of such complicated technique. Okay. And lastly, two conditions we have to read about, which is a renal calculi, stone or insoluble mass of crystallized salts, which salts of oxalates. So oxalate salts, crystallized salts, form stone or insoluble mass. They are formed within kidney. So these are also called kidney stones and renal calculi. And if they are present, then it might be possible that the blood will come out through urine. And if that is there, then again same thing. I'm telling you, if there is condition like kidney stone in the person, that will affect the urine condition, which means urine will also have blood in it. And once urine have blood in it, then clinically we can analyze first of all the urine, and then we can clinically diagnose that the person will have what? Will have renal calculi or kidney stones. Glomerulonephritis is inflammation of glomeruli of kidney. Glomeruli means plural of glomerulus. One more thing I want to mention is that whenever there is itis, arthritis or nephritis, then it means inflammation. Talking about summary, we have few important points to note here as well. That nature of nitrogenous waste formed and their secretion vary from animal to animal, and mainly depends on the habitat, availability of water. If the organism is living in aquatic environment, then it's possible that the person has either ammonotelism, or the person is not the person exactly, the animal or the organism is ammonotelic, right? Now, elimination of nitrogenous waste and maintenance of ionic acid balance of fluid is taking place by these excretory structures present in different animals. Now, DCTs of many nephron, I mentioned it very often, that DCT of many nephron join to form common collecting duct, many of which ultimately open into renal pelvis through medullary pyramid. So what happens is that DCT first of all join together to form collecting duct, and then from there it goes into medullary pyramid, minor calyx, major calyx, finally into renal pelvis, then into ureter, and from ureter it goes into urinary bladder, then into urethra, and then outside. And for removal for micturition we require two things. First, contraction of smooth muscle of urinary bladder. Second is the relaxation of sphincter, right? That is what was happening. You can read the micturition part. Then we have Bowman capsule encloses glomerulus, that is called Malpighian renal corpuscle, Malpighian or renal corpuscle, or Malpighian body also, and Malpighian tubule is specifically a excretory structure which is present in most of the insects like cockroaches, arthropoda. Now, filtration is a non-selective process performed by glomerulus using glomerular capillary blood pressure, GCBP, glomerular capillary blood pressure. Now, this very beautiful statement that will summarize everything. About 200 ml of blood is filtered by glomerulus per minute to form 125 ml of filtrate in the Bowman's capsule, which means that 1200 ml of blood is filtered by glomerulus, and this much filtrate is gone into the capillary, which means that blood entered was this, and blood going actually into the Bowman capsule from where reabsorption will take place and other things was 125 ml, of course. So 99% of reabsorption is taking place. That's very much evident because 125 ml of blood is filtrate that goes into Bowman capsule, and then urine, which is the product last at the end, what is formed is urine, and that's only 1.5 to 1 liter per day. And if we talk about the glomerular filtration filtrate amount per day will be 180 liter. So 180 converting to only 1 to 1.5 liter. You can see what's the difference. The difference is that 99% of reabsorption of filtrate takes place in different parts of nephron. So PCT, major site of reabsorption, selective secretion. If I recapitulate, then PCT is basically absorbing 70 to 80% of electrolyte, water, and all the nutrients, and it's also selectively secreting also. Then loop of Henle maintain osmolar gradient. Specifically, if I talk about ascending limb of loop of Henle is maintaining the osmolarity gradient and stuff within the kidney interstitium. DCT and collecting duct allow extensive reabsorption of water and certain electrolyte. Right, collecting duct allow reabsorption of water and also send urea, so it's kind of reabsorption only urea into the medullary interstitium. But there is conditional reabsorption of water and sodium ion only in case of DCT, that we saw that due to aldosterone there was reabsorption, conditional reabsorption of NaCl and water through DCT, and also ADH hormone also allowed later part reabsorption water through later part, which was DCT. So that was written clearly in NCERT that there is conditional reabsorption of water and other electrolytes or sodium ions or something like that in later parts of tubule, which was DCT. That is what I was trying to mention. And H plus, K plus, and NH3 are secreted into the filtrate, right into the filtrate, and then it will be removed out by the filtrate out of the body. That why is it done? It is done to maintain ionic balance of pH balance. Then countercurrent mechanism operates between two limb of loop of Henle and those of vasa recta. And then electrolyte urea are retained in the interstitium by this arrangement. Countercurrent mechanism not only in loop of Henle or vasa recta but also in their proximity. I hope this chapter is very clear to you, and especially revise this. Let me show you. This is very important, so please revise this, specifically the tubular secretion part, that PCT secretes H plus and NH3 and DCT secrete K plus and H plus, but mind you here K plus is reabsorbed by the proximal part. HCO3 is reabsorbed here because of ionic or pH balance. Here HCl is directly reabsorbed. As per the NCERT, what's mentioned in NCERT is primarily important for us, or it's primary thing for us. And here you have urea and water coming out, or there's reabsorption and other things. Thank you so much for watching, guys. Bye.